I'd like to, start by just saying that I come from Canada, and so I'm co-medical director of the Hereditary Cancer Program. And our program, our staff, we live and work on the traditional and ancestral and unceded territories of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh nations, the Lekwungen-speaking people from the Esquimalt and Songhees First Nations, and the Stolo people from the Musqueam and Sumas First Nations. And we give thanks for the opportunity to live, work, and support care there. I do have the following disclosures that do relate to what I'm going to speak about. Okay, I will give a bit of a background on genomic imprinting, but really quite rapidly try and take you into an overview of what we call parent of origin-aware genomic analysis, which combines two technologies, some that are in common use, long-read sequencing, and another one that is not so commonly used, strand-seq, single-cell template strand sequencing. I'll give you a bit of a summary of the validation that we've done so far and some examples of utility in hereditary cancer care, which is the world I live in. And so genomic imprinting is really, this is the understanding that really we require both sets of chromosomes coming from the maternal and paternal contribution for early development. And this really stems from early pronuclear transfer experiments, where essentially if you transplant the contributions from the egg, you have it's called gynogenetic, and you get no extra embryonic tissues. And then if you essentially transplant the contributions from the sperm, there's poor embryonic development. And so really when we look back in the timeline of what has been known about mammalian genome imprinting, these concepts were first established in the mid-'80s, and in the late '80s, early '90s, it was the discovery of the first imprinted genes. And really, so H19, IGF2, those are ones that people might be familiar with, those on chromosome 11. And really what this revealed was allele-specific expression controlled by epigenetic marks and thinking about DNA methylation. And so progressing in the early mid-'90s, it was linking it to human disorders, and in particular, a lot of them related to overgrowth or neurogenetic phenotypes. And then in terms of the late '90s and early 2000s, that was really establishing an understanding of that these were clusters of genes in these regions, and that were controlled by these imprinting control regions. And really these were set early on, and so with DNA methylation being established in the gametes, but essentially surviving the wave of demethylation that occurs after fertilization and persisting throughout adulthood. And then in the 2010s, it was considering the expansion of imprinting genes and thinking about how it worked. And that was really considering the long non-coding RNAs that we've heard about some already, and but in these ones, thinking them on the autosomes as well. And then really where I'm coming to is in the as the technology progressed, and so came the ability to start looking at these at greater resolution and thinking about technologies like long-read sequencing, where all of a sudden you can see not only sequence changes, but you can see these 5-methylcytosine epigenetic changes that are DNA methylation. And essentially, the ability to get a full sort of genomic landscape of what this looks like across all the autosomes, and get it in a haplotype-specific manner. And this, I just wanted to show this just to level set everyone on the life cycle of imprinting, and really, that this is established in gametogenesis, and really a lot of this work comes out of work done in mice in this understanding. But here you see in the upper one, you see the chromosomes, the maternal and paternal, one pink and one's blue. But then early on, there's erasure in gametogenesis. These are in the primordial germ cells when they're at the gonadal ridge in the developing embryo. And then there's reestablishment of the imprints that are in concordance with the sex of the fetus. But then these particular gametic imprints are ones that survive, essentially, the wave of demethylation that occurs after fertilization, and essentially persist throughout adulthood, as well as the consistent with the somatic methylation that comes as well. And there are a number of diseases that are linked with specifically imprinted genes. And in fact, this is not necessarily what I'm going to talk to you about today, but I wanted to just give you sort of an overview and show you some of them. One of them is transient neonatal diabetes. There's Beckwith-Wiedemann syndrome, and I'm purposefully not spending too much time here to get onto the other areas because Prader-Willi is one in particular, and Angelman syndrome, and also Albright hereditary osteodystrophy. But what I want to show you is that in consideration, like even though there are all of these particular imprinted disorders and that we learn as we have gone through our genetics training, in humans, there's actually these imprinted differentially methylated regions, at least one on every autosome. And so what I'm showing you here is 192 of them. In red is the maternal methylated, imprinted differentially methylated regions. And in blue is the paternal methylated, imprinted differentially methylated regions. So there's 148 maternal and 44 paternal that we've looked at and appear to be actually quite consistently able to tell parent of origin. So why is this important? Well, it's important is not only if you know about this particular region, it means that you can actually determine the parent of origin. And so it's useful in the particular syndromes where that region is affected, but also if you have another region somewhere on that particular chromosome, if you can actually link that region back and phase it with one of these regions to tell you parent of origin, then you know what parent it came from. So I'm going to show you a video that outlines our technology and hopefully gives them more insight.
A key challenge in understanding genetic risk can be identifying which parent a pathogenic variant came from. Besides the X and Y chromosomes, every cell in the body has 22 pairs of chromosomes, one from each parent. To determine the source of gene variants, we can use a method called parent of origin aware genomic analysis, which looks at differences between the chromosomes using only a blood sample from the person with the variant. This method uses two main techniques, long-read sequencing and single-cell strand sequencing, also called strand-seq. Long-read sequencing is used to spot differences between the sequences of the two chromosomes inherited from each parent. It can also detect methylation of DNA at selected regions, called imprinted regions, that differ between the parental chromosomes, allowing for the identification of the parent of origin. By aligning multiple long-read DNA fragments to a reference genome, we can construct larger DNA segments with imprinted regions, revealing the parent of origin for each segment. However, if the variant in question is far from these imprinted regions, long-read sequencing alone does not provide enough information. That's where strand-seq comes in. This technique involves looking at live cells to track the forward and reverse strands that compose the DNA double helix. Cells are treated with a marker that labels new DNA strands. After the cell divides, the new DNA strands are destroyed, leaving the original strands, one from each parent. By comparing sequenced forward and reverse strands to a reference genome, it is possible to figure out which genetic variants are inherited together as a haplotype on the same chromosome, allowing us to tell the parental chromosomes apart. Typically, 20 to 50 cells are needed to get a complete picture. By combining parent of origin information from methylation patterns obtained through long-read sequencing with the haplotype details obtained with strand-seq, we can accurately determine the parental origin of each DNA segment. This combined approach allows us to map out the inheritance patterns from both parents across all chromosomes, giving a clear picture of how traits are passed down through the family and which side is at risk of carrying the pathogenic variant.
essentially, means for the first time we can take a live or viable blood sample from a proband or an individual and tell the exact autosomal contribution that comes from the mother versus the father. And so we published this in Cell Genomics, and it's available if people want to dig into the details regarding it. For this, we use Oxford Nanopore long-read sequencing, but you can use PacBio because you are able to detect the methylation changes there as well. But really, just to summarize, this relies on there being viable cells, and it's really because StrandSeq is able to create this, essentially this shallow map of the whole genome that acts as a scaffold that you're able to essentially phase on the long-read sequencing that gives you the resolution of the variants, but also the methylation marks that you can determine those differentially methylated regions, and then therefore determine the variant parent of origin as long with all of the other autosomal variants on it. So we can do this for all autosomes because as I mentioned in humans, there is this ability to do it because of at least one imprinted differentially methylated region, although not every chromosome is equal, and so there are some with very few IDMRs and some that have more on them. In this particular case, in the Cell Genomics paper, we did it in 5 trios and had a 0.31 error rate for SNVs and a 1.89 error rate for insertions or deletions. Okay, so the world I live in is hereditary cancer, and that's what I think about. But there is this particular gene called SDHD, and it's part of the succinate dehydrogenase complex. And in fact, if you have a mutation in this gene, it does give rise to an increased risk of getting these non-epithelial neuroendocrine tumors if you have inherited the variant from your father, or if it comes through the sperm. If you have inherited from the mother, you don't have this risk. And it's not because the actual gene is imprinted, but it's because it's likely that there's an imprinted gene on the other chromosome 11, the maternal one, that gets commonly lost in these tumors. And that's the Henson hypothesis. Why it's relevant is we have a lot of times when we see patients and they just, for whatever reason, we cannot get segregation with the mother or the father. And so it's very unclear what sort of screening they should follow if they've come in it from not being affected with a paraganglioma. So there's a big rationale to get better precision medicine by understanding this and using this type of technology. And so this is just an example. It shows you, and this is the imprinting control region of Beckwith-Wiedemann locus on 11p15 in the maternal reads of the long-read sequencing that have been aligned to the StrandSeq. You can see that it's methylated in red, and the paternal one is blue. So that's up on the P arm, and the variant's all the way on the Q arm. And in fact, that's an entire haplotype, so you can tell that that's the paternal origin of that one on the lower part of that we're seeing there, and that in particular is an SDHD mutation. So we've seen this and seen that there's concordant predictions when we look at it in families. And in this particular case, I'm showing you data from 19 individuals, but we have done an additional 5 more individuals to show that this also holds true, that we are seeing concordant predictions when we look. This is 11x coverage on average, which is much lower than they usually like when you do this, but we're still able to define these. Okay, so why is this also important in other conditions? So SDHD is actually not that common, but what we get a lot of, so our program is essentially services the province of BC and the Yukon. That's 5 million people, and there's sort of over 150 different types of hereditary cancer risk. And we get a lot of referrals, the rates are going up, and our big cancer plan is to actually test every affected individual with cancer and then try and identify germline susceptibility. So when we identify a germline susceptibility like BRCA1, that has a frequency of about 1 in 150 in some populations, 1 in 200. In other populations, it's 1 in 40. But usually when we find it in clinic, we say that there's a 50% chance it came from mom or dad. And in this case, if we can apply this technology, it's actually really useful in terms of cascade genetic testing. This is a CDC tier 1 condition, like Lynch syndrome is another one, gives rise to colon cancer risk. And so in this particular case, it could improve a situation where we have very poor cascade genetic testing. So it's one of the biggest problems in hereditary cancer, and it's very well known that we have this problem. And it's, we have a publicly funded healthcare system, but still, like, even though there's no cost associated with the actual testing, not enough people get tested. And if people were tested for hereditary cancer risk, they could get options in terms of risk reduction, in terms of prophylactic surgeries if you can't screen for the cancers, or having increased surveillance. The issue with cascade genetic testing is that we do give a result to one individual, and we give them a letter, and they can share it with their family members, and then they tell them. And the problem is a lot of times they'll tell their kids, maybe their siblings, but not really tell their extended family members. But the risk in those family members is still, like 100 times the population risk, so it's so important. Cascade genetic testing rates are poor when we look in our own data, and we see it's 30% of first-degree relatives. And they're even worse in ethnic minorities, and we're not sure of all those reasons, if there's just differences culturally in how information is shared. But right now, the only solution in the field is to actually just directly reach out to people, and they call it supported direct contact. And it's sort of something like in the last 20 years, there haven't really been any good solutions apart from that one. And except, in my view, it's incredibly expensive to all of a sudden go out to both sides of the family. And so we have worked with our ethics team to basically create an approach to do this, except it is hugely resource intensive. And it turns out that people still want to tell their family members anyway, most of the time. We do think that segregation of the variant by using this information could help individuals. So what we've done so far is we've tried to validate it in different genes. So I told you about SDHD, that was chromosome 11, but we've also tried to validate it in other individuals. And so I'll give you data on 166 individuals with pathogenic variants and known segregation of clinical segregation of their pathogenic variants. And so these are in various genes, like you can see BRCA1, BRCA2. The Lynch syndrome genes are MLH1, MSH2, MSH6, PMS2, and EpCAMs related. And then there's some other genes that predispose to breast cancer risk and also GI cancer risks, or gastrointestinal cancer risks. We've tried to do this in an ethnically diverse cohort, just to try and understand what these marks look like, or methylation marks look like across the board. We've tried to get a good composition of male-assigned sex at birth, and also cancer history and age ranges. So what we've found is that we can only assign it, or we can assign it, I should say, in 90% of individuals. And so in 150 out of 166 participants, and this was across a range of chromosomes. The reason why we can't always assign it is because sometimes the imprinted differentially methylated region is not there. And so it's really an interesting phenomenon, and why it's so important to study different populations. It turns out in the European population, we can't see it in about 30% of cases. So when we do see it, though, we can get 99.3% concordance with known clinical segregation. So just one of the cases was discordant, and we're still looking into reasons why that is. So that we think the factors that can limit the parent of origin inference relate to homopolymers, and that's just in detecting the actual variant in the long-read sequencing data. Sometimes it needs manual review, or it's just a limitation of the technology. Sometimes there's some technologies that may be better at certain things. PacBio might be a bit better. And we'll have situations where there's rare phasing errors, or potentially a sample swap can happen, resulting in the wrong haplotype, but that's something that we have processes that are implemented so we can essentially identify those. And they can be solved by having further coverage with nanopore. And there's situations, and what you end up getting is you get discordant phasing. And so here in this picture, I'm showing you situations where you would want to see concordance of the actual IDMRs, because they're both supposed to be maternally methylated, except in this picture you're seeing discordance there, and so we know that there's an issue or problem. You can get runs of homozygosity, and so a lot of the phasing relies on there being heterozygous patterns that we can see, and so this is why it's really important to also do your validation in different population data sets, to, especially ones where there might be higher degrees of identity by descent. And then, and we know that there, in certain cases, you can get very ultra long reads that can help that out. And sometimes we do see that there's lack of allele-specific DNA methylation. And this is what I'm talking about, this issue of this polymorphic imprinting. So these are the challenges that we are seeing in our samples at the moment, but and despite those ones, we are able to have high concordance overall. There are considerations. So this is something, we're not used to doing this, at least in the clinical setting, genetic counseling, autosomal variants. Like, you don't usually consider being able to tell what side of the family it can come from. And so there's different considerations in terms of counseling. And those are ones that we're currently contemplating at the moment, and with ethics as well. But we have actually tried this in the clinic in some patients. And in fact, it's pretty interesting. So patients, they'll give the perception, they always thought it came from one side of the family, like my mom's side. This was a patient with unknown parental segregation of a pathogenic variant. And they had the variant returned sort of over 4 years ago with very limited cascade testing occurring only on the maternal side of the family. So we hope that this type of information can help patients sort of go after the side of the family. We are going to run a trial on this, and this is something supported by AstraZeneca, Merck, and CIHR, where we're looking at POGA-directed cascade genetic testing and combining it with a digital support tool for reaching out to family members, where they can mediate that sort of transfer of information and also give their relatives direct access to a portal where they can offer cascade testing. And we'll run it against the control of people just getting access to that digital portal. But what we're interested to understand is if it does move the needle on some of this cascade genetic testing. We do see the benefits to Canada, and essentially if you can save money on going after one side of the family, you could basically reallocate the resources to the side where we think that there's the highest chance at finding a mutation. The mutation detection rate should theoretically double, and you save the test costs associated with it. And theoretically, you would only have to do this at the branch points of the different generations. And of course, would help family members find a program in their area when we identify it. So we are doing, this is the clinical translation part of this, where we've got a health economist involved, we've got ethics involved thinking about it, and we're trying to get patient perspectives on this as well in terms of their views of it. So I just want to give these conclusions that genomic imprinting, even though we're not directly assessing disease-associated, like imprinted differentially methylated regions, it has significant clinical applications if you can infer parent of origin using it in potentially different regions. What we can see is that these imprinted differentially methylated regions are polymorphic, and they'll need characterization across the different ancestries. We are inferring that basically that POGA can accurately do this using only a blood sample, and that we are seeing a high rate of assignment with concordant prediction in clinical segregation in these individuals. It fundamentally changes the genetic risk assessment, is what we've learned, and it's a problem for me in clinic because it's just using up my entire startup budget, because patients always want to know, and I want to give it to them. But it does have the potential to improve cascade genetic testing efficiently. It's more accurate in its risk prediction. It has the potential to impact personal and clinical utility. So this, I've given you one example of one utility of this sort of powerful technology. But of course, if you're able to decipher these, full variants across the autosomes, there's more in there. And I think there's some really bright brains across the room. If you're interested in collaborating on this and looking into some of the data deeper, I would love that. First, I want to acknowledge my co-PI is Peter Lansdorp. So he invented StrandSeq back in 2012. Stephen Jones, he works on the long-read sequencing part. And of course, the hereditary cancer program and other important people, Vahid Akbari in particular, Lillian Cordova, and also the hereditary cancer program. We've got a big operation, and and again, there's uses beyond hereditary cancer. And with that, I'd just like to end and take any questions.